Alright, so I'm just going to talk about some of the physics of DNA, and that includes supercoiling and MEP, or molecular electrostatic potential. So what is DNA supercoiling? So we know that DNA in itself has a double helix, and this double helix is kind of like a coil in itself. So if we break apart the word supercoiling, right, it's coiling taken further, almost optimized for space, if you think about it. So you'll see what I mean in a second. So if I were to take this circular strand of DNA, probably found in some sort of bacteria or virus, and I folded it upon itself, that would be supercoiling. I'm taking that original double helix and I'm folding it upon itself. So therefore, I'm getting this optimized form of space so that there's less space, but the same amount of genetic material which is stored in DNA. So in other words, to put it simply, supercoiling is just taking that double helix and folding it in itself. And it doesn't have to be this figure eight. It can be any shape as long as it's compressed and stored and folded, and not expanded. So why does DNA supercoiling occur? There's two reasons why it can occur. The first reason is that during transcription, where we have our RNA polymerase come in and try and replicate our DNA, it opens up this replication fork, and this replication fork leaves behind a segmented piece of DNA which hasn't been opened yet. And this segmented piece of DNA experiences a force or a stress because it's being tugged upon individually, strand by strand, by this RNA polymerase in each opposite direction. So we can have an analogy for this in that if you were to take an elastic rope or some sort of rope and tie it up and pull on it like this RNA polymerase is pulling on it in opposite sides, then you would also experience a stress or force on the non-expanded parts of the rope, which are still twisted up. Now that's the first reason. The second reason is because of storage. Now, E. coli, or all the genetic material inside of E. coli, is approximately 3 times as large as E. coli in itself. Now that's pretty astounding. So what does this mean? It means that E. coli is using supercoiling to enhance the space that's inside of it for other cell processes. And so that's another reason why supercoiling can occur, and that's to store efficiently for other cell processes, but still retaining the same genetic material. Now, that earlier stress or force I was talking about, that's known as torsion. Now, torsion is the force an object feels as it's being twisted. Now, this force that an object feels that as it's being twisted can be broken down into two features, and that's shear strain and shear stress. Now, shear strain is, by definition, a force that causes the layers of the object to slide upon each other. So, in a way, although we can't really see it, while this object is undergoing torsion, this block, there is a sliding that's occurring from the original form that it was in to the form it is after you apply a force to make it twist. Now, shear strain, on the other hand, is that ratio of the change of this deformation to its original length. So, while before it was a force that caused layers of the object to slide upon each other, now it's that ratio of the new form of the object to its original form of being untwisted. Now, just some general components of understanding torsion. I'm not going to go too deep into these formulas, but I thought it'd be neat to just include these formulas for understanding. So let's say we had a rod, like before we had a block, but now it's a rod for simpler understanding. And we have this plane that goes between the rod. Now, if we apply a torque or a force, that's the type of force specifically that is featured in torsion. We apply a torque, what's going to happen is that this horizontal plane or line we drew is going to rotate. Now, after this line rotates, it's going to leave behind a plane in where it was before. And that angle between the original place or location to its final location is theta. Now, we can find that theta, because that theta will prove to be useful in a second. We can find that theta by this formula for angle of twist, and that's what it's called, that theta, angle of twist. Now, we can find that by multiplying the length of this rod times the torque divided by the shear modulus multiplied by the polar moment of inertia. Now, that seemed like a lot, but I'll just break it down quickly. The shear modulus is the inherent property of the material being twisted, pretty easy to understand, and the polar moment of inertia, just think about it as the moment of inertia as if you were spinning of an object, but now you're applying, it's fearing a torsion force. So it's moment of inertia for objects experiencing torsion. Now, we can find shear strain and shear stress earlier mentioned by multiplying the location of this torque times theta divided by L to get shear strain. And to get shear stress, we can multiply this torque times the radius at which this torque is being applied divided by the polar moment of inertia. Now, once we get this ratio found experimentally actually gives us the shear modulus. So this can prove to be very useful for not only manipulating objects with in real life that we can see, but also atomically, as I'm about to show you in DNA. So what scientists have found using technology is that this DNA, as it's being twisted naturally, is experiencing a torque as well. And that torque, therefore, the DNA is experiencing torsion. Now, it's experiencing torsion molecularly at each of its bonds. Now, these different bonds, these hydrogen bonds mixed with the covalent bonds, which makes up the sugar backbone almost in the DNA, what's going on is that these hydrogen bonds, coupled with oxygen and whatever the DNA is made up of, is that they're gyrating adjacent to each other and twisting through those covalent bonds. And with those covalent bonds in between, they're experiencing a torque as well. So as a whole, everything is rotating at the molecular level at these individual bonds. So here we see that there's 5 covalent bonds. Therefore, there's 5 torques. 5 torques applied to the hydrogen, 5 torques applied to the oxygen, and the covalent bonds. Now, after we apply this torque, and the DNA experiences torsion at the molecular level, the DNA is going to look something like this. Deformed, and we call this conformational change. So, scientists describe the supercoiling through parameters. Now, I'll just generally go over this, and then we're going to go into a theorem. So, first of all, we can describe it through 3 things: a twist, a link, and a rife. A twist is how many times one strand of a DNA is going over the other. So I like to think of it like a period of a function. How many times you're going to turn until you start repeating. So this length right here would be one twist, and when you go through another twist, then another twist, and so on and so forth. So this strand right here has one twist. The links, you can imagine as if there was an imaginary surface, and one strand crosses through that surface. So your link would be how many times your DNA crosses through that plate or surface I've drawn here, just for the sake of the presentation. And a rife is how many times each strand folds over each other, like I've shown in the middle here. So now we're going to go into White's theorem. What White's theorem does, really, is links the physics that we knew before with the biology we just understood now with the DNA. So what scientists did, by understanding the torques and the twists and turns that are responsible for the supercoiling, they found White's theorem. And so what this White's theorem tells us is that the links in the DNA, or how many times each strand crosses through that surface, is equal to the number of rifes plus the twists in a finite distance. And this can tell us a lot about the geometric nature of the DNA in itself, and can help us with some other formulas I'm about to show you now, understand how the DNA is experiencing that torsion. So with this, the time needed for that supercoiling to occur over a DNA strand can be given to us by this formula here. T sub d equals lambda squared over d. Now d is the occurrence coefficient. We don't have to really worry about that. That's calculated by understanding the friction between each bond in the DNA. That's also another physics concept there. And in the numerator here we have the length of the DNA strand. So this is something that can help us understand how long it takes to supercoil by also understanding physics. And then now we have molecular electrostatic potential. What this basically does is that it affects DNA by being partially responsible for its suspension. So like electrostatic potential in normal physics, like we see, it's also there atomically because of those charges being visible at the atomic level as well. And so in this case, torque is dependent on that polarity of the molecules. And through these maps, which are color-coded, the places where there's more red, there's more electrons, and where there's more blue, there's more protons. And with these maps and understanding molecular electrostatic potential, we can see how polarity and polar molecules affects the torque. So a more polar molecule might have a greater torque at which its axis is spinning between the hydrogen and oxygen. So that's just another thing that affects the torsion and that torque force that's being applied to the DNA. And so then we get to the importance of studying DNA supercoiling. I just briefly went over supercoiling and the electrostatic potential that's involved in supercoiling. So really, what studying DNA supercoiling does is it allows us to understand the storage of DNA in species that aren't humans. So what's been really common in the last decade was that scientists have been experimenting heavily with things that aren't humans, like E. coli, like I mentioned before. And when we experiment with these, we can extract certain genes from these certain viruses like E. coli and put them into drugs that can help us. So some things that E. coli is resistant to could be implemented in a drug, and we could eventually be resistant to that through these various pharmaceuticals. So this just opens a further gateway to understanding that storage in DNA. And another thing it does is it enhances our knowledge of genetics, such as DNA supercoiling has recently found to vary person by person. So just like DNA is genetic, that supercoiling and torsion is dependent on person by person. So the last thing I would say that's important about studying DNA supercoiling is that it interlinks disciplines that at one point weren't thought possible. So now what we have in today's day and age is physics and DNA/biology interlinking and showing that you can really unionize any discipline and show for the better what you can really accomplish with those two disciplines combined. Without physics, we wouldn't be able to explain supercoiling as much as we can now. We would say one DNA is folding over the other. We wouldn't have these phrases, torque, torsion, to help us truly understand the dynamics of what's going on at DNA when it's transcribing or being stored. So these are just some of the importances of studying DNA, and I just briefly went over some physics involved in DNA, which is supercoiling and molecular electrostatic potential. Thanks for watching.